Hi guys, I'm Christopher and I'm here with my colleagues. We are from the University of Ghana Medical School and we are here in the molecular analysis laboratory where we are going to conduct some molecular steps which is part of an ongoing project on microbial diversity. You know, Anopheles mosquitoes have been able to develop resistance to most of these chemical insecticides that we use for their control. Hence, it is imperative to find novel ways where we can be able to control these vectors of public health importance. So our idea was to look at the microbiome of where these mosquitoes breed. If possible, we can strategize by using push-pull methods or procedures of developing a new biocontrol for mosquitoes and also help in the reduction of the transmission of malaria. So I'm here with my colleagues, and from here we'll be moving straight to the bench to look at the procedure of how to look at the microbiome, the diverse taxons which are involved within these breeding habitats. So we'll be going straight to the bench where we are preparing for PCR, that's the polymerase chain reaction, to determine fungi and bacteria in the breeding habitats of these mosquito larvae. Thank you. As we come here, you can see this is Isaac. Part of the team member, and he has set up within the hood with his ice box containing both the primers and the master mix, and also some Epidoff tubes. He's getting ready to prepare the reaction mix which we are going to use for our PCR.
So, as you've heard earlier on, this is our hood. The reason why we are not working directly on the benches, PCR is sensitive. For that matter, you need a free sterile area to do it this way. That is why we are using our hood here, which controls all sterile environment as well. So here we have our ice packs. And then we have our component that we need for the PCR. So here you can see our primer, which is the 4R, which is the reverse, and then the 1F, which is the forward primer. As you can see them in the tube here, these are all working primers. They have been reconstituted. So starting, you don't go straight by fighting, but you need to do some few calculations to know the volume of your primer and then the DNA to use. So here we have some few calculations that we've done to be able to go about the volumes and then the concentrations of our primers to use. So here I have my primer. Volume of my primer to take is 0.25 microliters, which is of concentration 10 micromolar. So in starting, you have to first take your sterile tube, label it to be the master mix, because this master mix contains all components except the DNA template you'll be using for your PCR. So we label it MM, which means master mix. So we first go by adding our water, which is 45 microliters. this is our nucleus free water, as you can see. Others also comes in bigger size, like this. So depending on what you have in your laboratory, you can go with any of them. Whatever you are working on needs to be on ice. So this is our dream tag, and this dream tag contains all that we need, which includes the DNTPs, the magnesium chloride, and so on. This is my forward primer for fungi, it's specific. So this is fungi work as it's what we are doing. So this is our fungi primer, that is 1F. We add it to the mixture here. And then we go for the 4R fungi primer, which is the forward reverse primer. Sorry. So at the end of the day, we have our master mix, which we are going to allocate into the various tubes, depending on the volume you calculated. So this is our master mix, as you can see here. So I have up to 10 reactions, up to 8 reactions that I want to do. So with my PCR tube here, I then have to allocate 10 of my initial volume that I calculated for each sample. So we are going to add 5 microliters of our DNA, which was extracted from water sample. So once we are done, we go to our automation room where we have our thermocycler that we need to run our sample.
So this is where you have your thermocycler.
Yeah.
What does it do?
So this is a machine designed to provide the necessary conditions in amplification of the specific region of your DNA that you are investigating or gene you are investigating. So with this machine, it provides you with the necessary temperature that you need to amplify a gene of your choice.
So does it work similarly like in vivo in organisms?
I would say yes. I would say yes, because if you are working with a specific conditions in which is suitable for you or for the organism to optimize, then at the end of the day, whatever you are working with, being it the machine or the in vivo, tells you that it's the same principle, but could be that maybe one way or the other, just a step might differ.
Okay.
Yeah. So, because this is our first running, we need to program our machine.
Okay.
Yeah. And then because it's also the optimization system, we actually don't know which temperature will be suitable for our PCR. So otherwise, we need to include a gradient. And with this gradient, help us to know which of the temperature will be suitable for the amplification of that gene we are interested in.
Sure.
So as you can see, our target handling temperature should be around 55. And then the gradient of 7. So at the end of the day, you see a range of temperatures here. So we have 50.7, 50.8, 51.9 to 57.9. So this will be fine. If it doesn't work, you need to come back and then restart the whole PCR process again, and you have to alter these temperatures. That at the end of the day, you get the best optimization. So we have our program here. We've already programmed, so you can see our program is ready to run.
So how long will it take?
So depending on your number of cycles, this is approximately going to take us about 2 hours to done. Then we can continue with our gel electrophoresis. So we'll come back to continue the gel electrophoresis. Thank you. I'm Isaac.
Okay.